Hello, my name is Antti Vuolanto, and I'm the CEO of Herantis Pharma. And Herantis is about developing disease-modifying therapies to address Parkinson's disease and actually other neurodegenerative diseases as well. The necessary forward-looking statements, and then let's go into summary of Herantis. So our lead asset is called HER096, which is a peptidomimetic compound capturing the activity of a natural protein called CDNF. With HER96, we have great preclinical data demonstrating that yes, we are capturing the full activity of the protein, and we have demonstrated in clinical studies that we have good safety profile, but essentially great brain penetration efficacy. We are currently running phase 1b clinical study. We are already preparing for phase 2 studies. But what makes us actually really unique is the background of the HER96. As mentioned, it's based on a protein called CDNF, and HER96 actually mimics the active side of that growth factor. And we have already completed a clinical study with CDNF with fairly advanced Parkinson's patients, 17 patients with intracranial administration of CDNF. And in that study, we were able to see clinical benefits, we were able to see biological changes, we were able to see very interesting biomarker profiles. The biggest challenge was that CDNF as a protein had to be administered intracranially. And now we have HER96 that we can administer subcutaneously. And with HER96, we, of course, we want to have a long-term effect with disease modification, so slowing or even stopping the progress of the process of midbrain neuron degeneration. But what we have also seen in clinical and preclinical settings is a quick symptomatic improvement, which is very important for the drug development point of view. Parkinson's disease is an indication where we have an unmet clinical need. So the current treatments are able to treat the symptoms, but they don't actually affect at all on the disease progression. And the current therapies also have limited efficacy, so not all patients benefit from the treatment, or they have significant side effects. And as the disease progresses, at some point of time, the current treatments are not efficacious anymore, and nothing can stop or slow down the process of the neuron degeneration. So how we do that, how HER96 functions. Even though it's not known how or what triggers Parkinson's disease, we know quite a lot about the target neurons in the midbrain. And in those neurons, or in the surrounding of those neurons, there is a vicious cycle going on. First of all, we have their endoplasmic reticulum stress within the neurons. We have also accumulation of toxic alpha-synuclein aggregates, which is a hallmark of Parkinson's disease pathology. And actually, the aggregates are also going extracellular, causing neuroinflammation in the surrounding microglial tissue. And these activated microglial cells secrete so-called pro-inflammatory cytokines, which makes the life of these target neurons even worse. And all of this results in decreased dopamine secretion, and in turn results in movement disorder. But with HER96, we can actually we target the endoplasmic reticulum function. The target pathway there is unfolded protein response, UPR, so we are able to balance the proteostasis in the cells. And in preclinical settings, we have nicely shown that we are able to increase the survival of the neurons. We are able to decrease the amount of alpha-synuclein aggregates. We robustly alleviate neuroinflammation. And all of this results in increased or normalized dopamine secretion and very logically normalized motor function. So we are, in a way, very broad way addressing the Parkinson's disease pathology. About the data, we have really nice data set with HER96, and in this and this data has been prepared using a preclinical model that combines aging, alpha-synuclein pathology, and progressive disease. And in these graphs that you see here, the normal aged animals are the white, or the white bar represents them. The placebo-treated model animals are the red bar, and then the blue one is the HER96 treated animals. And now on the left-hand side, you see the effect on alpha-synuclein aggregates. And as you can see, the red bar is much higher than the white one. So the model is based on injection of alpha-synuclein aggregates. So it's not a surprise that in the model we have high number of those. But we can nicely bring that down with HER96 with treatment of 4 weeks subcutaneously. And actually at the same time, what we see with the neuroinflammation, again, elevated neuroinflammation markers with the placebo-treated animals, but actually very normalized situation with HER96 treatment. Very nice. But how about the dopamine system? So the first graph here represents the amount of dopaminergic neurons in the target area. And as you can see, we have 50% decrease with the model, but we can nicely protect the neurons by using subcutaneous HER96, which actually results in completely normalized amount of dopamine in the target area. And not surprisingly, this results in normalized motor symptoms, which is maybe best visualized with a video. So the first mouse here is just a normal aged mouse walking a bar walking test for motor coordination, no problems there. The second one has the induced pathology, placebo treated, and as you can see, the model animal really has strong motor symptoms. And then the third animal is the model animal treated with HER96 for 4 weeks. And as you can see, we have a very clear effect on the motor symptoms. The treated animal is actually even quicker than the normal animal in this test. Extremely happy about these results, and this preclinical data has been really well received by the pharma in our partnering discussions. Then into clinical data. So last year we completed the first clinical study with HER96, which was a single ascending dose safety study in healthy volunteers. First we had 6 dose cohorts between 10 to 300 milligrams subcutaneous injection in the abdomen area. Very clean safety profile. And then we had in part 2 12 elderly men and women from whom we also were able to take 1 single CSF sample, so as a measure for blood-brain barrier penetration in men. And the results are shown here. So in this graph, each individual point represents an individual. So we were able to take 1 sample at the predefined time point. And what you can see is that within the first 12 hours, we have a concentration between 50 to 100 nanograms per ml in these healthy individuals. And this concentration is matching with the concentration range that we have used in preclinical settings, providing good efficacy. So we are clearly within the therapeutic range. And this is very important data for us because the blood-brain barrier penetration is one of the bottlenecks in drug development for Parkinson's disease or other neurodegenerative diseases. And now we are running the phase 1b clinical study. We have actually completed already the first part, which is a single dose in healthy individuals to complement the previous data. Now we know how one single dose is behaving in humans, and we can use this data for designing phase 2 clinical study. We also had very nice safety profile. And right after the Christmas, we will start the part B. So we will recruit 24 Parkinson's patients, early to mid-stage Parkinson's patients in 2 dose cohorts, 200 and 300 milligrams. The study is placebo-controlled. Of course, mainly safety tolerability study, but of course important to study the pharmacokinetics of repeated dosing, study exploratory biomarkers, and also see if we have any effects on the signs and symptoms of Parkinson's disease. So we have a neurorestorative compound. We have easy route of administration. We already have very good safety profile. So we have very good position among the companies that are developing disease-modifying therapies for Parkinson's disease. Some words about markets and financing. So this slide summarizes the market opportunity of HER96. There are currently approximately 10 million people suffering from Parkinson's disease, 1 million or a bit more than 1 million in Europe and in US combined. We believe that there will be a market of at least 9 billion euros within these 2 main markets. This is for disease-modifying therapies for Parkinson's disease, and we believe that we have multi-billion market opportunity only in Parkinson's disease. And here is also good to remind you that HER96 and the target pathway that we are addressing, it's not specific for Parkinson's disease. We already have evidence with the CDNF growth factor in other neurodegenerative diseases, also in stroke, that we have there a good potential to utilize the same mechanism of action. About financing, I want to highlight the great achievement that the phase 1b clinical study is financed by a consortium of Michael J. Fox Foundation and Parkinson's UK with 3.6 million in cash of research financing. In addition to phase 1b, they support also some additional biomarker discovery work. Together with previously announced European Innovation Council accelerator financing, we have raised over 6 million euros of non-dilutive research funding during the past 18 months, which is of course great support for us. And we do have a commitment from EIC Fund for equity investments when we go forward. So currently we are running the phase 1b, we are preparing for phase 2, and we are seeking of course, out-licensing collaboration with pharma. And on that end, during the past 2 years, we have had meetings with 55 pharma companies, many of those face-to-face meetings. It seems that Herantis Pharma is very well known among the competition. We have personal relations with all the companies developing CNS therapeutics, and I think the discussions that we have been nicely progressing over the years. And good to remember that it takes always time to convince a pharma partner to make a deal. But of course, the objective of Herantis is to sign a major licensing deal, hopefully already based on phase 1b data, or if not, then we will start phase 2 and continue our efforts there. Yep, by that I want to thank or end my presentation, and happy to take your questions.
Thank you, Antti. Please have a seat.
Yeah, thank you.
Okay, so let's start with the current phase 1 trial. So in part 1 you have been progressing quickly and as planned, and now part 2 is starting with Parkinson's disease patients. So kind of talking about your expectations of this second part, are there any differences between Parkinson's patients and healthy volunteers that you would think that may affect the next stage? For example, are there known differences in pharmacokinetics, for example, that you are kind of expecting to affect the results?
There shouldn't be any major difference between the healthy individuals and then Parkinson's patients. It can be that actually the brain penetration is higher with patients, as it is known that the blood-brain barrier can be a bit leaky with patients with neurodegenerative diseases. But good to remember that the healthy individuals, we have given one administration, and now in Parkinson's disease patients, they will receive 8 administrations over 4 weeks. So the setting is a bit different. And of course, having 1 month treatment with 8 administrations for the patients is important stepping stone towards phase 2 clinical study, where we will anyway dose the patients with several months in order to really show the efficacy of HER96 in Parkinson's disease.
And will you be locking the dosing in this phase 1, or is there maybe some kind of, will you be testing different dosages at phase 2 or regimes of administration?
Yeah, that's a very good question, and we are currently collecting the data with the part 1 that is already completed with the phase 1b. We were able to collect information about how the one dose is behaving, and now we will confirm that with the patients, and based on those data we can then decide how many doses per week we will administer in phase 2. Most likely we are not going to test several different dosing regimes in phase 2, but we will fix that how many doses per week. But it's in a way the preclinical data plus the data with the healthy individuals plus the data that we collect with the Parkinson's patients, then we decide based on those data what is the dosing for phase 2.
One of the things that you're looking for now in phase 1 are the biomarkers. So if you think of the most positive case, so what can you get out of the biomarkers in the long term going to phase 2 and phase 3? What are the kind of most positive information and things you can get out of it?
Yeah. So on the biomarker side, we are concentrating on how HER96 affects the target pathway, which is a unique pathway for us. So we need to develop the biomarkers ourselves that are specific for HER96. And in an ideal setting, we would have a biomarker that has a clear change when HER96 administration takes place, or right after or hours after, so that we can see, we can demonstrate that HER96 actually hits the molecular target and does biologically what it is in a way meant to do. We are not trying to develop new disease biomarkers for Parkinson's disease, but specific for HER96, and that's important that we do that. And of course, if we would be able to develop such a biomarker, that would help further clinical development, but that would also strengthen our case towards partnering, because that's one of the key data that the pharma is currently looking at when they are evaluating targets for licensing.
You've talked about also here today about the possible other indications for HER96, including, if I remember correctly, metabolic diseases in your R&D day. So can you talk a little bit more about the other indications, and are there kind of practical steps to be accepted, for example, preclinical trials?
Yeah, so it's well known in literature that unfolded protein response pathway, our target, is involved in many diseases, neurodegenerative diseases, other CNS diseases, and also, as you mentioned, outside of CNS, cardiac, metabolic. Of course, the ambition of the company is to start studies in those indications, and naturally first step is to have preclinical studies, demonstrate a reason to believe, so that we could progress those to the clinic. But currently the company is concentrating on the Parkinson's disease, and we hope that at some point of time when we have potentially the licensing deal, we have more resources to open up these other indications, some of them which are pretty sizable ones as well.
In an optimal case, how long would it take from you to get from end of phase 1 to start recruiting in phase 2? What kind of time frame are we talking about?
Yeah, the ambition of Herantis currently is to have readiness for phase 2 once we have the full results of phase 1b. So most likely when we announce the phase 1b results, we first announce the top line data, and then it will take some time longer to have the full biomarker data. And also the biomarker data is important for the phase 2 design, so we need to have the full data set before we can, in a way, have the final design of phase 2 available. But presumably it will take maybe 6 months once we from the point that we have the full data set in our hands. Hopefully a bit shorter time, but there are many moving parts and variables that we need to take into account. But even 6 months is considering drug development, that's rather quick.
Yes, yes. And then in terms of funding, you have secured 15 million investment for phase 2 from the European Investment Bank. What kind of other parts for the funding do you need? Is it enough to get the partnership, or are there other routes than partnership deal?
Yeah, so the 15 million equity commitment from EIC fund is actually such a it's structured in a way that they can provide 1/3 of any subsequent financing round. So in a way, it's if we raise 45 million, they can cover 15 million. And from that 15 million, we have now covered 1.5 million in a financing round that we did actually exactly a year ago. So we have 13.5 million in a way left in that bucket. For the phase 2 financing, of course, once we are ready with phase 1b, we will discuss, negotiate with pharma, and then we need to decide whether we finance the phase 2 with pharma, or shall we out-license the asset to pharma that they will take care of the phase 2, or shall we finance the phase 2 ourselves? So there are multiple options that we need to investigate once we are ready to start a phase 2.
Okay, final question. As mentioned, there are some links between Alzheimer's disease and Parkinson's, and now the Alzheimer's drugs are leading now, and there's I think two drugs in the market. So have you heard any relevant news from that area now recently during the past months that are kind of relevant for Herantis or the development of Parkinson's disease? Are there any interesting news?
There are absolutely. So there are many similarities between Alzheimer's and Parkinson's, and one of the similarities is the accumulation of the toxic protein aggregates. In Parkinson's disease, alpha-synuclein; in Alzheimer's disease, beta amyloid plaques. And these two drugs that are approved, they address the beta amyloid plaques. So basically protein aggregate, toxic protein aggregates. And now if we think about our mechanism of action, we are addressing the protein aggregates, but we are also addressing the cell stress and inflammation, which in a way most likely provides a better way of addressing the pathology as such. And this is actually this is an item that we have discussed with the pharma, that they believe that what we have as a mechanism is more holistic and it's rather promising. And it seems like in Alzheimer's that even though these two drugs are approved, the efficacy is not great. It's a benefit, but not great, and there is much room for additional products. And we believe that we are in a way in a very good position that yes, if you treat part of the pathology, you can see benefit, but if you had a broader approach, how about that? And we are in that corner pretty alone currently within Parkinson's disease. So very strong position in that regard compared to the other companies developing disease-modifying therapies.
Thank you very much for the discussion, and best of luck for this final part of your current trial. So we will continue next with Faran Pharmaceuticals.